Thank you very much, Francoise. At this point, we move into the open discussion, which I think Francoise's talk has certainly given us a good start into. And if I could ask all remaining speakers from yesterday and today to join us on the platform here, and we will entertain questions. I have some questions to kick things off, and I'm sure we'll get going. This is the opportunity for those of you in the audience who have specific questions related to the science and also the ethical implications. And those of you who are watching online, if you want to send questions, please do so, and we will pick those questions up and transfer them to the front here. So please, everybody who's here, will join at the audience. I'm going to come and sit at the table as well. And I think I would just like to start by going across the panel and ask them to think about the implications of where gene editing is going. We've talked about human editing and germline editing. Let's come back to that. But where does the rest of the panel see the biggest impact that's going to affect society? How are we going to control the transfer of gene editing into somatic gene therapy, into agriculture, into things that can benefit society? But then, for example, when we talk about gene drive that will benefit and potentially prevent malaria, how do we control putting gene drive into other organisms that might suddenly change populations in the wild that we don't understand? So these are we're always in this kind of balance between the benefits and the challenges. So does anyone have a particular viewpoint on the other aspects of the impacts of gene editing? Go on, Rodolphe.
Sure. Well, I think this is a conversation that has been occurring for a long time. So it's not a CRISPR-based, change. This dialogue has been ongoing ever since molecular biology came into place. And we've heard yesterday and we've heard today that there's technologies that preceded CRISPR, like TALENs and ZFN and others. Certainly the gene drive conversation today was a great example of showing it's been occurring for a long, long time. But that being said, the dialogue needs to accelerate because the pace at which the science is unfolding and the number and the range and the breadth of applications, notably in ag, notably in food, notably environmentally, force us to have a dialogue with scientists, but not just with the scientists. And I think we've heard repeatedly over the last 2 days that we need to be comprehensive in who comes to the table, in whom voices their opinion, what kinds of true scientists or advocate scientists or groups of people come. And I think having a diversity of people at the table and having a diversity of opinions come in, including the public, is going to be the path forward. But that being said, if you look at what's happening right now, it is happening already. So the question is not what should happen or what is happening, is how we should manage where it's going.
Mm-hmm. So Francoise, Rodolphe just said that we are moving extremely fast and you were just advocating slow science. I understand what you mean by slow science, but how do we ensure that we have time to think and engage around the discussions of the broader implications of research? Is there a complete, is it impossible to stop and have slow science?
Well, I think from my perspective, the critical thing with slow science is it's not about stopping. I think the science continues, but what it does is that you actually create structures that support certain kinds of conversations, and so you make that a valuable part of what it means to do good science. And I just think that right now, and I did mean it, a lot of the rewards of science come in a very particular kind of way and domain, which encourage people to work in that particular way. And I do think that there will come a time when we are going to have to make some difficult choices, even about which areas of science to pursue aggressively. Those conversations are going to have to be happening with policymakers, and they can either happen in ways that, really people are confident that they've spent the time thinking through that and are going to make the best choices they can, versus making those kinds of decisions on the fly. And I think different people have found themselves in positions of power and authority making decisions on the fly. So I'm really trying to just create a different kind of culture for conversation.
questions from if anyone has questions in the audience, please can you go to the microphones, just get in line and we'll take those questions. So, in absence, keep coming. Okay, John, go for a question.
I enjoyed Francois' presentation, among others. And I was able to stay in that Washington meeting only one day, but I always carried away that poignant message by a lady in the middle afternoon who had a dominant hereditary disease. I think it was muscular dystrophy. And this is after the ethics discussion took place. And it was a heartfelt cry. She said, "You don't know what it would mean to eradicate this gene from our family's hereditary forever." So you're going to have a lot of that, and it's difficult to deal with.
Yeah. So I can comment a bit on that as well, because I'm after the gene editing summit, the National Academies of Science has taken a slow science step, I would say, in setting up a committee to really consider the whole implications of human germline editing. And we've been working on that for ever since the summit, and the report should come out in the new year. But I would say that during that process of having the committee and having a time to sit and talk to many people, we're seeing a shift in the overall mood of the conversation. And I think a lot of it has been influenced by the families, the people who are suffering from genetic diseases, to the point where the idea of a complete moratorium on germline editing, I think, is shifting. And I think that's a general, when I see out there, I think you're seeing that already people are beginning to think about where you would draw the lines, and knowing that in some cases you could have major impact on particular individuals and their families. So that's the societal, the issue of the society. Are we taking human hubris to the extent that we want to make genetically modified humans, or are we looking at the individuals who are being affected by these diseases, and we can provide them with a potential way of relieving that in their family forever? So there is a shift, and it is partly coming about from the slow science approach.
Maybe I'd like to comment on the point you made about having the structures in place, because as a community and public advocate, especially for the work that Paul is doing, this issue that you raised about the opportunity and commercialization issues is one that goes to the heart of what she's working on, which at the moment is seen as quite expensive kind of strategy that might be limited in application only in North American settings, when its real benefit especially is in a low resource country setting. But there's great importance in following the work that she's doing, because that cost of the strategy is continually being lowered by innovation. In fact, there was a great example of that work in gene editing for low and middle income countries just a couple of weeks ago. Having said that, I think there are great structures for participants and the people who need to benefit within the work that she's doing, because right from the beginning of designing and thinking about the trials and the implications of that, there is a great patient community on the design and further advancement of that work. And I don't think it's just a wish list. It's inclusion of people like myself and others in what she's doing that has really advanced that work.
Paula, do you want to comment?
Yeah, sure. Thanks, Robert. I think that's exactly true. I think in many ways you can look at what's happening in the sort of HIV/AIDS treatment arena and in the community, and really see, I think, a sort of a best practices about how society can move forward and think about these decisions. We're doing some very cutting-edge gene therapy. Gene therapy's been around for 30 years, and actually we have cured quite a few diseases. And the thing that's stopping us now is further developing that technology so that it's scalable and injectable and cheap. And the only thing that stops that is kind of time and money, because human ingenuity will for sure get us there. So I'm very excited and optimistic about the possibility for these medicines. We're seeing it today with kind of the huge advances in cancer immunotherapy. That's a gene therapy. That's curing cancers for which there are currently no treatments. I think very few people wouldn't look at the reports that are coming out about that and not think that was a good thing. So, I think, the technology is here. More importantly, the vision of what we can do is here. And now it's just a case of, being smart humans and kind of being engineers and figuring out how to do it. But as Robert said, I think the HIV community has always been, they've always been real leaders for, ahead of the curve for how society thinks about things. Just as they gave us the first ribbon, the red ribbon, I think they're also showing us how to involve patients in these conversations.
So if I can just offer a couple of comments in response, I think one of the things that's really important is that what I'm trying to communicate is two things. One of them, in terms of my own work, is I often use the metaphor of traffic lights with respect to the ways in which people can engage with questions in science and biotechnology. And on the one hand, you have a lot of people holding up big red signs saying, stop, 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 you're going to wreck the world. And another group of people with big green signs saying, go, 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 you're not going fast enough. And I think that with my comment about slow science, it's actually asking for amber, with the expectation not necessarily that it goes only in one direction, which is to red, but in some countries it actually goes from amber to green, right? It's telling you that you can now move off. And I think one of the things that's interesting that I'm asking people to think about is this line drawing exercise. So it's true that with respect to specific interventions, we have very important, active patient community groups, and individual researchers are often very good at going out to those communities and getting them to support some of the activities, et cetera. But we also have to pay attention to other people. The person I'm thinking of right now, a Canadian, is Gregor Walbring, who is a disabilities rights activist who actually worries about what he describes as the pathologizing or the medicalization of difference. And he actually has the term that he's coined, which is around ableism. Now, that may not in an obvious way apply to any one specific patient group like HIV. I'm not trying to suggest that there's an analogy there. But if you do take sort of the extreme positions of science that I'm looking at modifying the human germline, there is the claim that's being made that it actually belongs to all of us, not to a particular subset of a community, whether that be based on geography or underlying health indication. And so it's actually just an argument for broadening the community even beyond specific disease groups, to just say, what would it mean for all of us to have a conversation about that? And I think that there are recent past examples where people would argue that they've made mistakes. And I was at a meeting with OECD recently where a fellow from Monsanto stood up and said, we made a mistake in terms of how we thought we could just sort of, move forward with the science without, in fact, engaging and bringing along the community. And it's in that kind of a big picture perspective that I was meaning community consultation. And I don't disagree about, the fact that that happens with specific patient groups.
Last year, Mr. Clapper, National Security Advisor to America, said CRISPR is it is dangerous for American security.
So, yes.
And another slow science, I agree with Francois, because she knows Dr. Nancy Olivieri and the pharma company, Apotex. What is corporate media, what is corporate, she knows better than me. That's why I agree with the slow science. We must see who controls finance, and it comes to national security in this. So we don't know what will happen to CRISPR. It is important. Thank you.
So perhaps you raised the weaponization, and it came up a little bit yesterday, but do people want to comment on, everybody says this is an easy technology, it could fall into the wrong hands. And I know that the Department of Defense and so on is interested and concerned about this. So would people like to comment on that?
I mean, I think one of the metaphors that was used today of the hammer is a good example. And I like, likewise, the example of the pen. You can use the pen to write poetry and great essays and opinions, and you can likewise hurt people easily with words. So it's not a matter of the technology itself, because that's what scientists do. They're going to make discoveries. They're going to enable people to do certain things. This is the part of the biomedical research. You know, you have basic understandings of biological processes, and when you get lucky and you make a discovery that's enabling, you turn that into technologies that enable people to eventually, hopefully, do very good things with it, save lives. We've heard that over the last 2 days, and open new avenues of research and in cures. But because CRISPR is such a powerful technology, such an affordable technology, such a democratized technology, and because imagination is arguably the only limiting factor of what we can do with it, there are going to be people, and there are people that are thinking about negative uses. Whether it's fiction or whether it's real is yet to be determined. And I think people who work for the Department of Defense, by definition, want to be careful. Maybe it's not quite slow science, maybe slow policy, but you know, if you're going to make a mistake, you'd rather be too safe than too enabling. So I think this is where, when you have such a powerful technology, and it can be harnessed in positive and potentially negative ways, some people are more careful than others, more mindful than others, more safe than others. Maybe it's part of their job. But in the end, the job of most scientists is to focus on the science and try to be as positive and beneficial to humankind as one can be.
So that I think you fall on Francois's first page of Scientist by those comments. But I know that you do think about the broader aspects. But if science, I mean, and I think these kind of symposiums, obviously we're presenting the science to a broad audience. Many of us, and we have Fong Zhang actually coming back next week to give a public lecture about CRISPR, where we know that these broader issues will come into play. But how do we build the societal consensus that Francoise is pointing about? This is extraordinarily difficult to do and engage the right people. Anyone comment before I take the next question?
Coming from a food science background, we maybe in the 80s or 90s, we went through the GMO issues, and there was no consensus. I mean, if you're looking at the different countries, they regulated differently. So it's not going to be an easy task, really. And hopefully this time we know better, and so we'll be able to do that. But it's not going to be easy, that's for sure.
Right. Next.
Just a general question to the panel. Are there any lessons that can be learned when we look at this powerful technology and we look back at what's happened with nuclear power and how that sort of got ahead of itself in terms of the weaponization and now the backpedaling on trying to prevent proliferation, et cetera? Anything we can learn looking back at that in terms of putting the brakes on in certain aspects?
Anyone want to take that? Potentially.
I'm okay. Let's just dial back on the rhetoric here, because, we're talking about weaponization of CRISPR. All right. I'm guilty of, I think, putting a storyline in a TV show that was the first, bad use of CRISPR. We used it to kill a bad guy in a TV show called The Last Ship, season 2, if you want to go look it up. Sorry about the spoiler. The reality is we can't do that. To use CRISPR in a bad way in people, please, we can't even use it in a good way right now, okay? What are you going to do? Put it in somebody's drink, they're going to drink it, it's going to kind of go in and there's much easier ways to kill people, all right? So I just want to sort of jump in and say, yes, we have to kind of consider this, but I think it's probably more helpful to actually focus on what we can do and what we realistically are going to be able to do in the near future. Can we change subject?
So, still keeping on a bit of a maybe heavy note, maybe the last question. First of all, as an undergraduate student of U of T, I really appreciate this kind of panel to incite discourses, as you were saying, Professor. More of a question towards you. As you were saying, it's also important not only to look forward, but to look backwards in history. One of the things we can look at the foundation of the Royal Society, Francis Bacon was saying that the goal of science is power, ultimately control over nature itself. So does that, in any of your minds in such a way almost answered the question already of somatic and germline mutations in CRISPR that, well, that's the goal of science already.
Well, I'll offer up that I have a particular commitment or understanding of power in terms of the work of Michel Foucault, who ultimately says that power really rests with the masses, that if you really want to know who's in charge, it isn't the king, it's the people who allow the king to be there and to do whatever the king chooses to do in some sense. And so in large part when I'm talking about the democratization of science, it's because I actually do believe that at the end of the day the power will rest with the people, so far better that you should have a well-informed public such that they can in fact hopefully exercise that power wisely for the benefit of all humankind. I mean, I sit here in a room believing that everybody that is a scientist or an aspiring scientist wants to make the world a better place. And the interesting thing is we're going to have different ideas about how to do that. But because I believe we have that goal in common, what we need to do is we need to help people to understand how it is that the science can help take us to that better place. And you know, in response to the other comment, I mean, I think that it is important to recognize that when I talk about exercising the moral imagination, I do mean to do that in a way that's responsible. And it isn't just science fiction for the sake of science fiction, but I think it's worth thinking back to some very interesting novels which have come to pass in some way, shape, or form. And so it's just interesting to have those kinds of conversations, not because we want to get there, but that because it will allow us to set ourselves on a particular path where we know that we'll develop the science in a particular way that we can in fact move forward to the goals that we all want to achieve.
So just before the next question, I just want to turn back to Paula. I agree with you in many ways, but I do think that it is not just interesting, but perhaps important to try to imagine what the future looks like, because we are moving so rapidly, and things that are not possible today will be possible tomorrow. If you turn back the clock 10 years and put yourself there and see where we are today, you would never have imagined the kind of things we can do today, and that's just a very short period of time. So yes, we can't do it now, but as part of the broader debate, I think we do have to look beyond the bounds of where we are today. Jim, you want to go, and then I'll...
Sure. So I'm going to change the topic, and it's really stimulated by the talk this morning by Sylvain, where he, I think, re-glorified the role of bacteriophages. And it's great to see somebody so passionate about bacteriophages. But I wanted to remind, and I don't need to remind him, that the biotech revolution basically started from understanding bacteriophage. And the question is here, can we, it is a great example, can we reinvigorate the interest of our politicians to understand that we need to focus on fundamental science without asking questions which we know the answers to already. In other words, focusing on applied science. And if so, I know on this table there are lots of ambassadors for that point of view, but you've got a fantastic opportunity here. And I think it's cyclical, right? You go through from fundamental discoveries and then applications, and then you think, oh, let's get more applications. But I think this is now is a really good time, and I'd encourage everybody. But Sylvain, especially as, when you talk about your discovery grant from NSERC and the challenges of NSERC and CIHR and the other agencies, not just in Canada, in recognizing the importance of basic science. This is perhaps an opportunity to reverse that trend.
Yeah, absolutely right. And I was actually quite surprised last March, the Quebec elected officials even had a scientific breakfast. So we met with politicians and talking about CRISPR, obviously, but talking also about increasing funding for basic research. And one of the things that I kind of raised at that time was that even when we sequenced the human genome back in the days, this was supposed to solve everything, right? And I know I was a student back then, and well, it did not. And now we've got this tool that is pretty, and Janet was talking about benefits. And really, I think the benefit will be to understand our own genome. Really, if you look at a genome, there's so many genes and proteins, we have no idea what they do. And now we've got this tool to to be able to go and study them and try to understand this fantastic network that is a human cell or any kind of cell. So yeah, I think this is a great opportunity, first of all, to kind of sell the story, but also to encourage politicians to increase funding. And which I was really surprised, though, last March, is they're really open about this. They just, yeah, we need to kind of help them, obviously, but no, it's a great, yeah, I think it's a great story, yeah.
If I can expand upon this, so I think again that over time there's been multiple examples, and CRISPR is the most recent of basic discoveries where it comes kind of out of nowhere, right? Putting the puzzle together. We talked about that for the last 2 days. So basic science will oftentimes yield at a low frequency great technologies, but a concept that we heard about today was opportunity cost. So if you have a finite pool of money, whether you're a company, whether you're a government, whether you're for-profit or not-for-profit organization, you have a certain limited set of funds to allocate to advancing or solving a problem. And I think the key here is to balance that portfolio, because the success rate for basic discovery science is not the highest. So it is important to allocate funds to do this, and to not make assumptions as to where the next great technology will come from, because we don't know. This is the whole point of doing this. But at the same time, we can't just do that, because we need to make sure that a bulk or a significant portion of the pie is de-risked, the opportunity cost is not so high, and that you can keep advancing what is tangible, what is almost there, and then apply those technologies that came farther out and bring them to the clinic, like we've heard yesterday. So I think the key here is to balance. Just like we balance our opinions when we talk about ethics, I think it's important to balance the risk and the investment when we talk about research.
Summer, is that an online question?
Yeah,
or your
I am
question?
representing Ravi Valenki, who is on our webcast. Thanks for tuning in. He wanted to thank you all for your thoughtful lectures, and he's wondering if you can comment on 3-parent babies and the possible ethics around it. Thank you.
You want me to take that, or are you going to take that?
You go first.
So 3-parent babies, do people know what 3-parent babies are? So this is the opportunity to try to treat embryos that have mitochondrial disease, so disease that is encoded in the mitochondria. And in that case, if the egg the nucleus, the DNA that comes in from the egg and the sperm is okay, the defect is in the mitochondria. And because your mitochondria are inherited from your mother, essentially, if you could replace all the mitochondria in the egg with wild-type mitochondria, you would be able to produce a baby that was genetically related to mum and dad, but would not carry the mitochondrial disease. And so the concept is to transfer the nuclei from mum and dad in the egg into a wild-type egg that has normal mitochondria. And this is there has been a first report. It's not fully documented, I have to say, but there has been a first report of a baby born that way. And this has been another big question along the way, because the argument is that this is, in fact, the first germline editing. So you are actually changing the mitochondrial DNA going into the future. The NAS had another committee looking at that. The NAS is very busy and came out with a rather wishy-washy conclusion. But in fact, in the UK, this has now been listed as a potentially doable approach for patients with mitochondrial gene therapy. And the first report was actually not from the UK, but from another group. So it is probably the easiest and certainly technically doable approach to curing a genetic disease in the germline. Does not involve CRISPR-Cas at all, and not necessarily so.
So that's a use of technology which I think is actually really interesting in terms of at least engaging with the question I was trying to raise with respect to public education and also opportunity costs. So in terms of what's important from my point of view with the science is that it's a potentially effective intervention if the woman has mitochondrial disease that's actually caused by a defect in the mitochondrial DNA. And a lot of the mitochondrial diseases are actually due to defects in the nuclear DNA, so that wouldn't even be a treatment for any of those couples. But I actually have a paper coming out on that, and I'm only mentioning the paper because I don't know the numbers, I don't remember them, but I do the math in the paper, and I argue that if you take just the United Kingdom and the United States, which are the two places that have issued reports, the United Kingdom saying that this is acceptable to go forward with this in a clinical context. The US having said it's acceptable to go forward provided you only transfer male embryos in order to avoid some of the concerns about heritable modifications. So I take those two populations, which is about the two countries together is about 400 million people, and I show that you would actually have an impact of less than 100 healthy births per year. And so I do end that paper asking a question about the opportunity costs. And the reason I make it in that way is because the argument for going forward with this intervention has all been about reproductive choice, not about social justice, and not about basic knowledge. I end the paper saying if you had built an argument in support of mitochondrial replacement or human nuclear genome transfer, which I think is actually the more accurate term, on the grounds that this is really important in terms of basic science, I would have been far more supportive, and I think it would have been more honest, because I think that's why we should be doing that science, not because we're going to help 100 healthy births out of a population of 400 million, and that's just looking at the numbers for two countries. So as I said, I have a paper coming out, and I'm remembering the numbers off the top of my head, but really that's what it amounts to.
I, in fact, don't disagree, Francoise. There is no basic science, particularly to be learnt from those experiments. And actually, this is a further argument around the germline editing, because most of the initial ideas that people have about what one might potentially be able to use germline editing, nuclear editing, around are also very rare occurrences, genetic disease. So this... I think I've told other people, at some point I have felt sitting on the NAS committee that by focusing on human germline editing, we are focused actually right now on a problem that will be only applied to a few people. It'll be expensive, very few numbers of families. However, there are, that's why I say you should look to the future, there are future things that will change our ability to do human reproduction. Last week, there was a paper in Nature showing, or Cell, I don't remember, one of those journals, basically showing that you can take a skin cell from a mouse, turn it into an iPS cell, and turn it in culture without any, putting it back in animals at all, turn it into culture into a perfectly viable, fertilizable oocyte. So you can make eggs in the petri dish. And people have already shown that you can make functional, not sperm, but spermatids that you can then inject into the oocyte. So it's only a matter of time before that comes together to essentially do mouse reproduction in the petri dish. We are not there with humans yet, but that doesn't mean to say it's not a future coming down the pipe. So if you're making stem cells into gametes, you can genetically modify stem cells. You can really change the future of the human race. So this is not here today, nor is it likely to be here in the future, but this is why we do have to exercise our imagination.
I just have a quick question. So this is leaning more towards the ethics side of the discussion, as I'm not that scientifically educated as a grade 9. So if I do say something that's related to CRISPR and Cas tonight, I'll probably say something stupid and I'll look stupid. So
no stupid questions. There are no stupid questions.
So I was just thinking, would it be a good idea if you were to include the general public and raise public awareness about clinical trials in CRISPR, how it really works and how we shouldn't expect a cure overnight, which could risk the loss of many donors and supporters of organizations and events like Run for the Cure?
Who wants to do that? Somebody want to comment?
I feel really strongly that when I talk about working on gene therapy and what I'm working on, if I didn't believe 100% that it was going to lead to a cure, I wouldn't do it. And so when I tell patients and donors and grant funders that I'm working to cure HIV/AIDS, I completely believe it. We'll get there. So to me, it's not a lie. It's not a It's not misinformation. I mean, I think, it's just, this is what we're doing, people. We're really smart. We've got amazing technologies we're developing. The rate and pace of change is extraordinary. And there's amazing people working on this. And I think, quite honestly, calling it anything less than that actually does a disservice to the people who are afflicted with these conditions and the passionate, smart scientists who spend all day working on this stuff. They're cures.
Manuel, do you have a comment?
Maybe I could bring a European perspective to the discussion. So the meeting in Washington was initiated by the American Academy of Sciences together with the Academy of Sciences in China and in the UK. And I think there was a representative of the Leopoldina Academy of Sciences in Germany, but in principle was pretty much organized by the US, having less strict regulations than in Europe, whereby in Europe you have this Oviedo Treaty that specifies clearly that one should not modify the genome of human germlines. However, surely the modification of genomes of human somatic cells, as long as it is for research and development purposes or preventive and curing purposes, that this type of modification is allowed. Now, having said this, I just also would like to mention that I don't think that one should be afraid of CRISPR-Cas, at least not now. I think the technology is very good for research and development. As it was discussed, I think there are other ways for bioterrorism. I mean, I'm a bacteriologist. Bioterrorism, in the sense of spreading bad viruses of bad bacteria, have been a concern for very long years, and yet I don't think that there is really an example of an epidemic linked to bioterrorism, as I think it is also easier for anyone to play around with chemicals than to play around with CRISPR. I also think that with regard to the slow science and fast science, surely CRISPR is evolving as a technology very, very fast, and it's very useful for research and development purposes, and also for the medical field in an indirect fashion via research and development. But I believe that it's actually very slow, and it will be slow with regard to a direct application as a direct tool, just because of the drawbacks of the delivery technologies. And also, as Paula said, and also in the context of the fact, as it was discussed, that here we talk about the direct treatment by CRISPR-Cas9 of a low number of human genetic diseases that are relatively rare. And I believe that it will take a number of years before one sees the technology successful in clinics, if ever, but we hope all that it will be successful. Having said this, I also believe that the technology is not ready not ready yet for genome modification in human germlines in a safe manner. So in a way, I think it goes very fast. But for what we are discussing about today, I don't think it's going to happen in 1 or 2 years, which is very good because it allows to start discussions around the subject, as it was mentioned. And in Europe, I witness in France and in Germany a lot of discussions going on involving policymakers, involving politicians, involving bioethicists, philosophers, experts in sociology, scientists, and so on. I participate in some of the discussions, and I think the strong will of having those discussions and increasing the level of those discussions with really incorporating, as you mentioned, the public as well, is really that at the point it goes fast, but there is still time to engage discussions, to have the time to really think carefully of how we would like to use the technology, knowing that it takes always some time to establish some laws and policies. So this is what I wanted to say.
So let me go back briefly to some things that Janet said before and in Francois's talk and others have said. And just in the context of if we don't understand history, we're destined to repeat it. So we've talked a little bit, or people have talked a little bit about stem cells, the GMO debate, but we haven't really heard the outcome of those in relation to the present discussion. And I'd like to go back a little bit further to the mid-70s, and that's the recombinant DNA debate, which in modern molecular biology was probably the first such public debate, the Asilomar Conference and the results from that, and similar things, that is germline modification was certainly part of that discussion. And I guess I'd just like to ask, what have we learned from some of these particular Asilomar? So people of my vintage who lived through Asilomar and know a fair amount about it would have said, as we've discussed today, that if this were repeated today, the public debate and the debate at Asilomar was very important, but I think many of us would have said that the outcome may not have been what the outcome would be today, that is particularly a moratorium, and we've heard a little bit about that. So my question is really, are we reinventing wheels, but what have we learned from previous debates on these topics?
Well, I'll offer one quick comment, which is that when I was first approached and asked if I would be on the committee, and when I heard that David Baltimore was going to be the chair, I asked the question point blank, is this meant to be, another Asilomar? And the response I got to my question was no. And I think that that was the right answer insofar as from a process point of view, I don't think you can have Asilomar again, just from a process point of view. That was a closed meeting. It was almost exclusively scientists. The reporters that participated, all of that information was embargoed until after the meeting came, et cetera. And I think that's no longer possible in the world that we live in. And so in part, it's in recognition that we don't have conversations in those ways that I want to actually try to foster high quality conversations. And maybe I didn't, maybe that didn't come through as clearly as I needed it to come through in the presentation. But that's actually what I mean by slow science. And I tried to say earlier, it's not about stopping the science. It's about saying, while that's happening, can we have high quality conversations, and can we trust the public that they are in fact educable? And by that I mean respectfully engage with the things that people don't understand, so that they aren't saying, well, I don't know if I can ask a good question. All questions are good questions insofar as if you have it, probably there's at least one other person that has it, so it's worth answering. So I think for me, that would be my first response. I think from a process point of view, an event like Asilomar won't work. It has to be open, and given that it has to be open, we ought to engage respectfully with people, and that's why I keep talking about both science literacy hand in hand with ethics literacy, so that we can have discussions about what's the right question to even engage with. And I take it to heart. If the right question isn't, let's not talk about the militarization of this because that's just so far down the pike, but let's talk about this other thing because it's actually up close, and that's great because then we're actually having a different kind and quality of conversation.
Thank you for the.
Oh, so I think one there's another set of lessons that has been learned ever since that meeting, and I think scientists now understand they cannot do it in a vacuum, and they can't just do it in a vacuum with other stakeholders that are ancillary, but also with the public. And you mentioned the word educable. I think educable and educated are important criteria, and I think nowadays you see scientists, you see universities. You have forums, access to the public, where it is part of the scientist's job to educate the public, to reach out to the public. It's part of the Gardner Foundation's mission to make science and scientists accessible so we can engage in those conversations. We can engage in this dialogue. We can try to educate. Sometimes the scientists are the ones educated as part of that process when they talk to the public. And the second point is that in today's world, Asilomar 2.0 could never happen. because we have, live tweets, because we have the internet, because we have, freedom of information. And it cannot happen, likely will not happen again. And I think this is where, history teaches us lessons that is up to us to kind of, abide by and be mindful of. And I think this is why we're here. This is a great example today of fielding questions from, anyone who has access to the internet today. And we will address the question no matter what. So I think it will not happen again, but we also have to have those lessons from the past. And I think this is where the GMO conundrum and the GMO history and the food and ag part of the equation are very important, because we know people are very concerned. But we also know they need to be educated about the advantages and caveats of that technology.
Dan?
I just want to add something on this, is actually something that really struck me during Francois's talk is, often from scientists, we tend to say that public needs educated. But I think one thing that struck me during Francois's talk is that I also needed, as a scientist, to be educated in that process. And, just the concept of slow science, gave me a headache when I was sitting on my chair. And, I need to understand a little more about it. And I think what's very important is that as scientists, we're not sitting there thinking that the responsibility is only from the other. I think it needs to be, we need to internalize these issues as well. So.
Okay. We're running out of time.
Yeah, I just would like to say again, as Rodolphe mentioned at the beginning, CRISPR-Cas does not raise new questions. I mean, those questions have been asked for decades since we know how to recombine DNA. But surely the technology is versatile and allows, at least in the preciseness and in the versatility, to modify the genome in a broader fashion. But what is important to mention is at the point, it's a better technology because it's a more precise technology. But I think that all those questions are also to be seen into the context that we talk about CRISPR. But as you mentioned, we talk also a lot about other technologies around CRISPR that are also allowing to somehow consider human germlines differently. So it's not only about CRISPR, it's also about all the technology around CRISPR, which are, new technologies of delivery, the possibility to work with stem cells, the possibility to derive germlines from stem cells, etc. So this is what I wanted to say.
Okay, I'm going to take the last question.
Great discussion. Thank you for that. It definitely points to the importance of further research into designing and engineering safety switches before this technology is brought to the clinic, particularly within the germline context. But I do have a much more general question, and perhaps unrelated, but for the young scientists in the audience, what advice might you have for people who are starting their graduate studies, looking into which way to direct their graduate studies, how to select graduate supervisors, that sort of thing?
Oh, that's a big question for the last question.
You can come to my talk at 4 o'clock.
There you go.
Room 2173.
Got it?
There is one thing I want to say with regard to CRISPR. As I mentioned at the beginning of my talk, it's really changing the way biologists can ask questions, because now you have really a technology that allows to ask questions in types of cells and also creating models of diseases that were not really possible before. So I think it will allow to expand a lot research that is cross-disciplinary, and that is really research merging different types of areas in biology to understand new aspects of biology.
Okay. Well,
Okay.
Sorry.
Yeah, just to go back to your question, actually, lucky you, really, to start your graduate studies when all this craziness is going on. I think also something we tend to forget is we're training students as well. You know, you're very lucky to be, familiarized right away about these ethical concerns and questions. And I know when I was a student, there was no way this was discussed at all. And I think you guys will, will learn about this, and you'll be our best ambassadors, going to your family, your friends, and talking about the technology. And that's kind of important too.
Okay, well, I think it's been 2 great days of science, and I think it's been a very interesting discussion this afternoon. I'd like to thank all the panel members and all of you who've raised the questions and really stimulated debate. So, thank you very much. This is the end of the Gairdner Symposia for this year. We will be back again next year, and in fact, as I said, we have other symposia that take place during the year, and we're looking to have more public outreach and other events. If any of you would like to be on our mailing list and hear from us, not every day, please just drop your email over with Summer and Paige over there, and we'll put you on our list. So thank you very much. Enjoy your weekend.